I'm Cassidy Gentry.
I'm Harriel Peters.
I'm Paige Strucker.
And I'm Haley Royal.
And we're so excited to be sharing our Cell Limit project with you guys. The title of our project is I'm Cultured, and our experiment was designed to test multiple widely used products, cleaning products and antibiotic products on bacterial resistance. We started our experiment by going around the Meredith College campus and testing multiple highly touched surfaces like tablets, tables, phone screens, computer keyboards, and door handles for bacteria. We transferred the bacteria that we collected onto these plates here that contain multiple concentrations of our substance solutions to see which substance bacteria would become resistant to the fastest. Also, how and why they became more resistant to some solutions over others.
We hypothesized that the bacteria would gain the most resistance to the hand sanitizer. We were not incorrect in this statement, though we did find that the bacteria had a lot of resistance to chlorine. We used Clorox to test it. At one point on our highest concentration of Clorox, there was 100% growth coverage on the plate. It sounds scary, but it's not surprising. The surfaces we took the bacteria from are constantly being cleaned with Clorox, meaning that it's more likely to gain resistance. Hand sanitizer also didn't work because we tested it with the hand sanitizer Governor School East gave to us on the first day we were here. This hand sanitizer was only 65% alcohol when you need at least 70% for it to be effective.
So just a little rundown on bacterial resistance. So when these bacteria are exposed to certain substances like antibiotics and these cleaning products, some of them will die, but some of them will live because of mutations that they have in their genes and their plasmids. And then these bacteria will reproduce and pass on these mutations, and then we end up with a whole lot of resistant bacteria. Also, one pretty crazy fact is that some resistant bacteria can actually pass on their resistance genes to surrounding non-resistant bacteria that are in the area. So kind of crazy.
So these are actually the plates that we use to test all the bacteria. We put all our solutions on the bottom of it, and then we took the bacteria that we swabbed from all over campus and we put it on the solutions, waited a couple of days to let the bacteria grow, and then we looked at it. So this is the ampicillin. This is our best or our most working solution, and it pretty much has no bacteria on it. This is the hand sanitizer. This is the same hand sanitizer that we got in the beginning of opening day to use to clean our hands. As you can see, there's a big difference in bacteria, which is really not a good thing. And this is before using the hand sanitizer and using after the hand sanitizer. Before, after, and that's not a good thing. And there's actually really similar results with chlorine, as Paige said earlier, and that is because, as she said earlier, we do clean our hands, we do clean surfaces, especially since Corona started. We've all been cleaning things more. That bacteria is literally growing resistance as we speak. The more we use it, the more bacteria is growing, resistance and trying to fight it off so that it can grow and thrive better. So, we were given, as we use hand sanitizer, don't use that to clean your hands completely. I mean, the hand sanitizer that we use only has 65% alcohol, and it's better to have more. But please wash your hands instead. That is the best solution that you can use. And if you are going to use hand sanitizer that has at least 80% alcohol, or you could just use use alcohol itself. Our science teacher actually doesn't even use hand sanitizer. He just carries around alcohol with him, and that's always an option.
All right, some solutions that we have, because a lot of people are like, what can I do to help? As a community member, like Harriel was saying, try and find hand sanitizer that's at least 80%. Because alcohol is a solvent, it can dissolve those bacteria and hinder them from reproducing. So that's why the concentration has to be so high. But in the medical field, if all these bacteria are becoming resistant, how are we going to be able to treat infections caused by bacteria? In the UK, they're actually studying phage therapy, and that refers to bacteriophage, which is a type of virus that naturally can kill bacteria. So if it's injected into you, it's not going to harm you in any way, but it's going to target those bacteria and it's going to kill them off. It's not currently in the US today because it's still being developed and researched by scientists, but hopefully it's something that we can move to in the future as a solution to the bacteria resistance. So yeah, Thank you guys so much. We appreciate it.
Thank you.
Bye.
Project Planesthesia was centered around the research question, can you anesthetize a plant? And so for that, we looked at three main things. Would petroleum ether serve as an anesthetic? Would an anesthetic slow down the rate of photosynthesis within the Venus flytrap? And would we be able to observe any external changes? And so one thing is that I want to give you some background that Venus flytraps are not carnivorous plants. They just use the nutrients from the insects that they trap in order to help their photosynthesis process. Also, to explain a little bit of the mechanism, there are trigger hairs within the lobe of the Venus flytrap, and if two of those trigger hairs are touched within 30 seconds, then the Venus flytrap will entrap the insect only partially so that small insects can get out and so that it won't entrap an insect that's also too large. And after an hour or so, this seal becomes more airtight.
Okay, so some background chemical information. We use petroleum ether, and this is actually a byproduct of gasoline known as benzene.
Alright, so these are the materials that we use. So right here, you can see the CO2 and O2 rates. This is how we measure the photosynthesis and to make sure that the plant was still alive and if it was asleep or not. And so here are the O2 and CO2 sensors that we have right here. Right now you can see the photosynthesis rate of this plant. And then if you can see here, there's also a fume hood that we would use. So we would put that fume hood over the container just to make sure that anesthesia wouldn't go everywhere.
So for our actual procedure, we did two experiments over 2 days. In our first procedure, we exposed the plant initially to 8 milliliters of petroleum ether, and we added 2 milliliters of petroleum ether every 10 minutes. Also, every 10 minutes, we tested the Venus flytrap by triggering one of the traps, and if it closed, then we continued to add the petroleum ether. After about 6 hours, the traps were still closing, and at that point, we had added 22 milliliters. So we decided to come back the next day and modify our procedure a little bit. Instead, we thought that maybe exposing the plant to a high amount of the petroleum ether for a smaller quantity of time might be successful in putting the plants to sleep. So we immediately added 20 milliliters and took a time lapse over 60 minutes. And during this time lapse, we observed that the Venus flytraps actually slowly closed on their own without being triggered. And so we concluded that that was kind of a natural defense mechanism in response to being exposed to petroleum ether.
Alright, so here we have like our main 6 observations that we recorded. So right here, this is our control plant. This is before any petroleum ether was given. We just wanted to put this here just so you could see the before picture. And also, it was really interesting because there was an insect caught in here, so there was a very pungent smell. So 3 days later, we exposed the same plant to 1 milliliter of petroleum ether. We really didn't notice any change, so we upped it to 4 milliliters. And again, there was really no response to our external stimuli. However, we did notice some droopy leaves.
And then down here, these first 2 pictures show the observations for procedure 1. So there's a before and after. You can see that in the second picture, after the plant was exposed to 22 milliliters, the traps were very droopy, and overall the plant just was not in good health. And then this last picture, it was taken 24 hours after the second procedure, where we exposed the plant to 20 milliliters. And that plant is healthy and is actually sitting in that container right there.
Alright, so our end result, overall we concluded that we did not get an external stimuli like we had initially hoped. However, there was still an external stimuli presented when the plants did get droopy leaves. Internally though, there was a change in CO2 and O2 rates. What this means is that the carbon dioxide and oxygen levels within this tank increased, showing that there was no photosynthesis immediately after petroleum ether was introduced to the plant.
Thank you.
Hi, welcome to the squirrel house. The original plan was we were going to motor power a squirrel and use Bluetooth on our phones. So we were going to use a motor like this one that we currently have powering a fan, as you can see here. So that works, and we were gonna use that to move our squirrel, but it took us a lot of time to work on this house because none of us had any prior experience on any of the programming or the circuit stuff. So we spent a lot of time on the house and didn't get to the squirrel until just yesterday, but that's all right.
So in the house we have our Arduino over here being powered by this 9-volt battery. And so how the Arduino works is it's like a miniature computer and it's storing a code. And so this code is reading through the sensor, and it's basically telling the sensor that whenever there's an object within 35 centimeters, then it'll send a signal up through this relay, which is like an electrical switch. And so when that relay is powered, it goes from being normally open, meaning that there's no electricity flowing through the circuit, to normally closed, meaning that the circuit is on. And so when it changes to normally closed, it tells this battery to send power into all of these lights, turning them on. So when Varun rolls the squirrel in, the lights turn on.
So when it's less than 35 centimeters, right here, all the lights turn on.
Yeah, that's pretty much our project.
Hi guys, I'm Kayla.
I'm Lisette. I'm Chloe.
And I'm Grace. And we did our project on water quality. So our main question was, what is sustainable in terms of water quality for the different organisms that use the water sources on campus? So we're looking at temperature, pH, dissolved oxygen, and turbidity. So for temperature, we found that it's an overarching factor. So it affects like vegetation on top of the water and the species that you'll find in the different water sources. For our dissolved oxygen, we found that it correlated with the temperature. So if you have a high temperature, you have a low dissolved oxygen, and vice versa. For the dissolved oxygen, that's important for like the different organic material in the water and the bacteria because they really like to munch on that stuff. For the pH, we wanted to look for that just to see, hey, is there like acidic precipitation? Is there runoff that could be affecting our indicator species? Because they're sensitive to certain toxins. For turbidity, we're looking at that factor just to see if the water was cloudy, if there's different minerals and sediments in the water that could be preventing our indicator species from eating what they want to eat because that can really affect the predation.
The indicator species that we decided to use were the Cope's gray tree frog and the squirrel tree frog. Both frogs are indigenous to the region and they're also found all over Meredith campus, so we decided that they would be a good way to measure the water quality. We also chose them because of their permeable skin. That would mean that they're more sensitive to toxins and chemicals, and they'd be a good measure of the quality of the water. So what we did is we used the EPA standards of what was sustainable for frogs, and we compared them to the data that we found. We found that in general, frogs survive best at about 75 to 85 degrees Fahrenheit, but they can survive at lower temperatures of around 60 degrees Fahrenheit. We also found that the turbidity has to be below 30 NTU in order for frogs to survive. We found that the dissolved oxygen level has to be at about 0.7 to 6 milligrams per liter, and we found that the pH has to be at a neutral pH of about 6.5 to 7.5, but frogs can survive at an acidic pH of about 4.
To go through with the procedure, some of the materials we used were lab tests and other tools. On site, we took temperature and dissolved oxygen. These were factors that would vary if we took them back to the lab, so that was a limitation that we faced. Another thing is that we took turbidity and pH while in the lab, so to do that we collected samples. These two on the left and these two on the right are from the fountains, so they're a lot more clear. And we collected samples from the quad fountain. Behind the oaks, we went to the left pond, right pond, Little Lake, and the Science and Math building. We also documented it on the campus map, and we tried to get as much of the campus as we could, so that we could have representation. Some of the pictures we took show the turbidity. So on the left, we see the right pond behind the oaks before precipitation versus after. You can see there's a high turbidity before versus after there's really low turbidity.
Okay, moving on to our data and conclusions. So compared to what Chloe said about our research, what would be most optimal for our frogs, we found that with temperature, the frogs would survive best in the quad fountain after rain, the left pond after rain, and the right pond after rain, which we can see in these comparative bar graphs that we made. The blue being the before treatment and the red being the after treatment. For our turbidity, we found that all these fall within our standards of below 30 NTU and actually fall within the standards of North Carolina for lakes and reservoirs without trout, which is below 25. And we even did standard error bars for our multiple readings for these values, and we ended up finding In regards to our factors between precipitation and no precipitation, these were kind of inconclusive because it decreases after rainfall for the Quad Fountain, but increases with the left pond after rainfall, and it's just very all over the place with that. And the same thing happened with our dissolved oxygen, where it was like increase after rainfall, decrease after rainfall for certain bodies of water. But in regards to what would be favorable for our frog friends, we found that the left pond behind the oaks for both conditions, the right pond behind the oaks for both conditions, and the little lake for after rainfall would all be okay for our frogs. In regards to pH, all of the bodies of water, no matter the treatment, would be okay for the frogs, but the only really suitable one where they would thrive would be the little lake behind the oaks because it's the most neutral. From this, we found that more natural bodies of water, such as the little lake, the right and the left pond, were better suited for our frog friends. The science and math method being the worst, so don't put any frogs in the science and math method pattern. And we found that after precipitation typically produced the best conditions in regards to turbidity, dissolved oxygen, temperature, and pH. But it was a close race, so that's not super decisive. Some areas for growth that we wish we could do, we wish we could have looked at floating systems as well as these lentic systems. And we wish we could have taken a wider sampling so that we could have been more inclusive and more representative in our data of the whole population. And we also wish we could have been more controlled with things like when we went out during the day and the temperature and the overall conditions of when we took our samples. So that's it. Thank you so much. Thank you all. Bye.
Hi, my name is Nikki Corsi,
And I'm Max Nelson.
and our presentation was testing how CO2 affects plant growth. So what we did for our presentation is we made 2 of these greenhouse type structures, as well as we had a control group. So we started off with having 3 plants. We had grass seeds, fast growing seeds, and bean plants. And we wanted to test how different concentrations of CO2 affect each of these different types of plants. So like I mentioned, we had 3 different environments. We had a controlled environment, which was pretty much just regular air out in the lab. So that's like right here in the picture. And then we had a medium CO2 group, which we added 50 grams of dry ice to, which is about 20 liters of carbon dioxide. And then we had our high group, which is that group, and we added about 100 grams of carbon dioxide to that, or 100 grams of dried ice, which was about 40 liters of carbon dioxide. And then over about a week period, as the plants were growing, we measured them and saw how the carbon dioxide affected how they grew.
So as you can see, these different sorts of plants are affected differently. This graph shows bean growth with the different concentrations of carbon dioxide. So as you can see, the high concentration of carbon dioxide here in red, the medium is in the orange and the green is in yellow. You can see that with the higher concentration of carbon dioxide, we see greater heights and greater growth in the beans, which leads us to believe that carbon dioxide is possibly associated with a greater growth rate. We see the same things in these Wisconsin fast plants, where the highest concentration of carbon dioxide is the highest growth, and the medium right in the middle, and our control is green down the bottom, is right at the bottom. So this also leads us to believe that it's positively correlated, there's a positive correlation between higher levels of carbon dioxide and greater and faster plant growth. Now we did notice that this wasn't the exact same for the grass. As you can see, the grass has even a little bit of a negative correlation. As the carbon dioxide goes up, the plant growth actually decreases. Now, we think there's several reasons for this. It's possible, one, that our method of administering carbon dioxide through dry ice may have stunted the growth of these plants if the grass was particularly temperature sensitive. The colder dry ice may have decreased the growth. It's also possible that because different plants deal with carbon dioxide in different ways, the growth speed and the number of stomata as well as the size of the stomata could have resulted in different rates of growth.
So our greater significance of this project is we kind of wanted to see with global warming and rising greenhouse gases and carbon dioxide in the atmosphere, how are plants going to react to this in the future? And generally speaking, we think that plants will probably grow faster due to these greater carbon dioxide levels. But like we saw with the grass, it really does depend on the type of plant as well. That's our presentation. Thank you.
Yeah, thank you.
So, alma mater, this is Emily Saxon, we're circus, and our our project was to make a heating and cooling system. This right here is the temperature sensor, and it senses the temperature in Fahrenheit and Celsius and the humidity of the air. Right now, the temperature is too cold, so the heating system is on, and that's what the clicking noise is. It's on a delay, so that the coil doesn't get too hot and burn through stuff. But when the temperature gets too hot, then the fan will come on to cool down the temperature sensor. And these right here are relays, and they help with telling everything what to do and when to do it.
Our system is fully automated so that based on like the value of the temperature, the code that we programmed decides whether to turn on our heater or cooler. Like right now, because it's cold in this building due to AC, our heater, which you can feel, is heating up the air around this. But once it reaches a certain temperature, that value on the computer is going to tell the program that it's reached too high of a temperature and the fan's going to come on to turn it on. So this is just like kind of a fully automated heating cooling system. The only thing, like, we wouldn't need to be here. The only thing we need to do is change a battery every once in a while.
It is essentially like your AC and heating unit in your house, except more rudimentary.
I'm Cameron Green.
I'm Lily Brickman.
I'm Maya Sinclair.
And I'm Shay McDowell.
And we did our project on hybrid DNA. We compared different hybrid plants around campus. We compared sweet bay magnolia, big leaf magnolia, and southern magnolia plants found around campus, as you can see in the map. We also compared 4 different types of tomatoes that you can find at your local grocery store, such as U.S. Roma, Mexican Roma, golden cherry, and cherry tomatoes to find the genetic differences between the hybrid plants.
And when we're talking about hybrid plants, basically what we're talking about is just 2 different plants cross-pollinating, and it's a resultant fruit or seed or plant that comes from that cross-pollination. And something that happens a lot in hybridization is what's called polyploidy, which is when the hybrid has multiple sets of chromosomes, and that can cause issues, especially with infertility. And that can be used commercially, like seedless watermelon is an example of infertility because there's no seeds, and that's something that's desired commercially.
The main goal of our project was to see if the commercial tomatoes that we buy from the grocery store are really tomatoes, or have we bred them over and over to become something that is more palatable to us. So our project was mainly focusing on gel electrophoresis. What we did was we extracted DNA from our tomatoes and from our leaves to try to run a gel on them using a gel like this, where we ran an electrical current through it, and we were able to try to separate the DNA. So when we did our extraction, we crushed up our tomatoes and we extracted them with an enzyme that would separate them and make them easier to see. Unfortunately for us, when we put our DNA in our wells to run our electrophoresis, the DNA didn't show up as well as we desired. So there is a small dark line here underneath one of our wells, which was the only DNA that showed up, but we weren't able to compare our DNA with our tomatoes since none of the DNA showed up outside of that one tomato. What we did learn was that we need to add more stain to our tomato DNA in order to help us see our DNA clearly when we run our gel, and this way we will be able to compare them directly.
And while we would have loved to have been able to run our DNA again, time is definitely a restraint, as well as our resources like funding. So if we had more time and funding, we would definitely run our DNA again, and also choose a wider variety of plants to test and even make our own hybrid plant, which would be great. But because of, again, time, we weren't able to do so. And from this project, we've concluded that while it is possible that hybrid and non-hybrid plants do have very notable genetic differences, besides those that are visible, like size and color, we can't conclude that from our study alone since our DNA was inconclusive.
Thank you.
Thank you.
Hi, my name is Simran.
I'm Shivani.
And my name is Hoy.
And our project is on the environmental differences between GMOs and organic plants.
Some of the questions that we asked when we began this experiment were, what's the true difference between GMOs and their organic counterparts? So how do GMOs truly affect the environment around them? So pH of the soil, pests that they attract, etc.
Now, before we get into our setup, we want to talk about what exactly GMOs are. GMO stands for genetically modified organisms, and these organisms have had specific alterations to their genome for various purposes. In an agricultural lens, some examples of genetic modifications include quicker growth and a natural pesticide. As this is a new development in agricultural science, there's a lot of controversy around it, especially with the health implications of it.
So our setup, you can sort of see it here. These are the exact plants that we put outside in the natural environment. And these are how we tested the pH of the soil. And these here are the seeds that we use. These are Wisconsin Fast Growers. These are organic, and these are GMO.
The reason we chose soybeans in Wisconsin Fast Growers is simply due to time constraints. They both grow very fast in a short time period, and obviously we only had a very short time to do this. Additionally, they are widely available. They are very cheap and easy to be delivered here. And we chose to do it in the natural environment rather than a controlled environment because in agriculture, things are typically grown in a controlled environment. So we simulated the typical conditions, and we also wanted to see multiple different variables, including pH and the presence of pests.
So our main hypothesis was essentially that we believe our organic plants would exhibit a lesser bug presence than their GMO counterparts, whether that be bites taken out of leaves, eggs, or even insects crawling around. However, they wouldn't grow as quickly.
Some of the materials that we used, again, you can sort of see everything here. The only two things that aren't here are the herbicide that we use, it's just a spray, and the potting soil that we used.
As for our procedure, it was just like growing any other plant. We planted them in soil and we watered them and we placed them just outside the greenhouse along the Meredith line.
So here we have some snapshots in time. These would be the GMO soybeans, so these plants right here. It took a bit longer for them to germinate compared to the Wisconsin Fast Growers. However, as you can clearly tell that they've grown much taller than these over here. About a week ago, there was a stretch of about 5 to 6 days where it was nothing but 90 degrees with little cloud cover. And as Simon will get into in a moment, we have reason to believe that actually caused the wilting and in fact death of some of our Wisconsin Fast Growers. Some issues will be brought up along with that. However, the GMO soybeans didn't exhibit as bad of a decrease in plant growth as the graph on the right. There was only a slight decrease in productivity. And as you can see here, towards the end, it begins to tail off in vertical growth. That is because the plants, rather than going straight up as a new sprout, they now begin to leaf and grow outwards more. Their rate of growth is also much more proportional to each other compared to the radicism of the fast growers. We also wanted to know what kind of bugs were in the area. So although we couldn't exactly dig up every single potted plant that would in fact kill the plant, we had this tinfoil tray, not dissimilar to this black one right here, albeit with a solid bottom, where there was water, and what bugs would do is they'd fall in, they wouldn't be able to get back out. And so we have pictures of some pill bugs, moths, scarabs that had fallen in there, so that we could see exactly what sort of bugs were in the area.
As Sway said, the Wisconsin Fast Growers did start to wilt, and we have various explanations for why that is. First off, the Wisconsin Fast Growers, although GMO, specifically were altered with the fast-growing gene. This could potentially cause a rapid growth that the environment cannot support. Additionally, they weren't equipped with any natural pesticide while the GMO soybeans were. This shows how important pests play in the growth of a plant. Finally, as Soy said, there are multiple different environmental factors that can affect plant growth, including heat and very heavy rainfall that can put pressure on the plant.
So as Simran mentioned, the herbicides are actually built into the GMO soybeans genome. However, in the rest of our plants, the herbicide was not built into their genome. And to test our theory that the herbicide is required in the genome of the GMO, we sprayed our organic soybeans with an herbicide to see if pests would still be attracted. And as you can see, they did not grow, so the pests did stop their growth. But that's also where the controversy in GMOs come from. Herbicides cause many health effects in humans as it's a toxin, and it builds up in our liver. Over time, that could cause respiratory failure, kidney failure, etc.
And to go further in as to why the organic soybeans didn't grow, when we tested the soil acidity, we found that the organic soybeans had a higher soil acidity than both the GMO soybeans and the Wisconsin Fast Growers. So some potential explanations is that the organic soybean death caused soil acidity, or the other way around, soil acidity caused the death of the organic soybeans. Additionally, as they are organic, they had no genetic modifications to make them more suitable for the environment, like growth magnification and a natural pesticide.
So before I get into the issues of our experiment, some of the things that we did well was the fact that we measured all of our plants in millimeters, all of the water that we watered them with in milliliters, so our measurements were pretty exact throughout the experiment. Some of the issues is that we did only get to monitor these plants for 2 weeks, so if we were to do this again, we'd do multiple groups of plants over a longer period of time. And we'd also test it in multiple locations, including one indoors as a control to compare our outdoor groups with our indoor groups. Thank you.
My name is Chili Song.
My name is Gerald True.
My name is Rachel Davis. We were trying to find the biodiversity of microorganisms using samples from the pond water and soil samples from the pomegranate tree, fig tree, oak tree, reeds, and moss. We took the soil and we screened it in the process. Compared to the initial process, the soil had like a greater much surface area, and the water collected was about 50 milliliters to 100 milliliters. We took a droplet of that and looked in the microscope to find the microorganisms.
So for our findings and what we were able to figure out from this information that we gathered, we had a few key ideas. First, that of the terrain and soil samples, that the pomegranate tree had the most biodiversity and the most number of organisms down here. We also had that the soil samples were without algae, which was the primary component in the water sample at the pond, to show, photosynthesis is pretty much not happening under the soil. And also another key feature is that the ciliated protist, the paramecium, it's an aggressive, fast-moving, very in size protist is the most common. It was found in every sample, and it was pretty much the biggest predator out there that was able to attack and move through everything. It seems that the cilia around it made for the most efficient means of travel, while also allowing it to hunt without having to fear being eaten because it is so fast.
We had this presentation that we made. We photographed over 40 different microscopic organisms of over 30 different species in all our research findings. And we identified the organisms by class and species. And then we made a presentation to depict what they look like for others to kind of get a visualization of a microorganism, because it's not that common to see a depiction of them.
Yes, and we even included an interactive microscope station so that people would have the opportunity to try and find some with samples from namely the pomegranate soil, as it was the most biodiverse here.
Hi, I'm Katie.
I'm Brody.
I'm Stella.
I'm William.
And our project is called Hydrate or Dihydrate. And we, when we first got to campus, we heard a lot of people complaining about the water from the sinks tasting bad, and they didn't want to drink it because there was rust around the sinks, and they thought it would make them sick. So we decided that we put that to the test and see if it was a placebo effect or if it was actually not good for you. So we ordered some test strips, and we decided we were going to conduct a blind taste test to figure that out. And our hypothesis was that it would be a placebo effect.
So we tested 5 different water sources from around campus to determine if it was based on water from the city or to determine if it was based on the source of the water in campus. So we tested each source for 16 different parameters, and we tested each of them 3 times to determine that, to make sure that there was no inconsistencies. The only inconsistency that was found was that the cafeteria water had higher levels of chlorine. But other than that, all of the tests were the same. So the 3 tests, each time they had higher chlorine, and all the others were completely fine.
So then we wanted to conduct a blind taste test with a lot of participants. So we set up a table on the quad, and we brought forms out, and we brought our 5 sources out that we had previously tested, and we labeled them 1 through 5 so that the
Participants would not know where each source was from and would not have any bias. And we had them rank the sources 1 through 5 from the worst tasting to the best tasting. And while most people could not tell which water source was what, we did get an overall best tasting and worst tasting water source. And we found that the best tasting water source was the smart water, the bottled smart water. And the worst tasting water source was the cafeteria water. And we think that maybe the cafeteria water might have been the worst tasting source because of the elevated chlorine levels that we found, maybe giving it that pool-like taste that some people may have ranked it the worst tasting.
To continue our research, we tested water from 3 outside sources, which were the town of Cary, Lake James, and Orange County. Everything came back exactly the same, except the town of Cary had higher levels of chlorine similar to that of the cafeteria. From our blind taste test, we had 2 participants from the town of Cary who each thought that the cafeteria water was their favorite source of water. So we think that your hometown can influence what type of water you will like wherever you go. However, because we only had 2 participants, we cannot prove this for sure, so we would like to continue studying to see if there is a correlation between this. It also debunks the theory that the dorm water is not safe to drink, as it has the exact same chemical makeup as all 4 other sources tested. Thank you.
Hi, so for our Silema project, we tested the biological effects of stress. Now, after a hard junior year for most of us, it was really difficult adjusting and to the pandemic especially. And of course, we were all plagued with stress. And it was actually Tucker's idea to look deeper into the effects of stress and how our body reacts. So around the board here, we have different adrenaline molecules. Well, actually, they're all the same. But they're just hormones that your body releases when you're in a stressful situation. So we had a few questions going into this experiment.
Some of these questions consist of being how do people react differently to different stimulus, and the other question was how does your body respond when you're exposed to certain stressors?
Yeah. So we actually, one of the cool things and lucky things right now is we actually tested right here in this spot for about 5 hours in total, testing on 20 different subjects. Now we had a whole entire procedure to go along with this. Grady, you want to?
So we got our sample through a Google form, filling out, trying to get as a random representative sample of the students at Governor School. And we tested them. We decided on 2 variables that we could choose to determine their stress levels through an EKG monitor to measure their heart rate, and respiration monitor belt to go around their chest to measure their breaths per minute to determine the respiration throughout the experiment. So in terms of stimulus, we chose a slideshow to show them images and sounds in that slideshow, as well as an ice bath where they put their hands into an ice bath to elicit a physiological response and measure their stress levels from those stimulus. So in terms of measuring through an EKG, we measured a lot of raw data, and then we looked deeper into the wave. So this is one example, a good example of measuring through an EKG. So this is the same subject, but through here, this is our control data. At the beginning of the experiment, we measured 1 minute of control data, and we determined that their resting heartbeat was around 56 beats per minute based on the distance of the wave. And then this is their heart rate while their hands were in the ice, and their heart rate was around 111 beats per minute. So you can clearly see in this 2-second interval, their heart rate severely changed due to the stress.
So this is really interesting. Where in most cases people would show an increase in heart rate while their hands were in the ice bath. Occasionally we would see some subjects who would have a decrease in heart rate while their hands were in the ice bath. And we didn't understand this information for a while. And right here is the first time we really saw this. Participant number 8, they put their hands in the ice, and we observed an average heart rate of about 55 beats per minute, which just perplexed us. And for days we didn't understand until we looked at the respiration graph. Now this is the same subject right here. And at about the same time, we noticed a severe decrease in respiration and their respiration rate. And we concluded that the participant was holding their breath. And that was a really groundbreaking discovery for us. And we learned that people have different responses to different stimuli, where some people would hold their breath some people would breathe more rapidly, and these are just different bodily responses to maintain homeostasis or anything to keep warm to conserve energy. And in some case, so that varied about 75-25, 75 being most people would increase their breath, increase their respiration, their heart rate would increase. Now, in one case in particular, 1 out of the 20 people we tested, they showed no increase or decrease in heart rate, no dramatic increase or decrease in heart rate. And we still do not understand how this person varied this much compared to everybody else. We would like to test that person if we had more time.
We also took a lot of observatory data just to kind of see if there's any sort of contrast in how somebody feels that they react to stress versus how their body will subconsciously react. We asked each person throughout the test that for each stimuli, they gave us a ranking on how stressed it made them feel. And something that was really enjoyable to see was that a lot of people would give a certain rating that was lower on the stress scale, whether they gave it like a 3, kind of saying that it didn't stress them out. But looking at stuff like their EKGs, we could see a really high increase in heart rate, and we could see them taking a lot more breaths, just kind of showing that their body is reacting in a way that it feels stressed, even though they may not recognize themselves to be in a stressful situation. We also had, before and after the experiment, a questions sheet for them to fill out, and one of the questions on it was seeing how comfortable or how stressed out they were before the experiment, and then seeing how they felt after the experiment on a level of comfort and stress. And it was cool to see that 60% of people actually said that they were more stressed out before we even began the experiment versus after completing the entire test. We feel that this kind of ties in how anticipation actually affects the way that we feel stressed, and that naturally, as we assume the worst, just because as beings we don't like to be caught off guard or be caught by surprise, that we normally anticipate and prepare for the worst, that our stress will actually build up in pure preparation for that.
So I'd say we go over the rest of our interesting findings, and Alec, would you like to? So one last piece of information is before each subject was tested. Before each subject was tested, we would ask them if they had any fears or phobias that we should be aware of. And for one subject in particular, they told us that they had a fear of bugs. And during one of our images, I'll let Greg take over in a second, but during one of our images, we have a picture of a spider. And when the spider came on the screen, our participant rated it a 10 out of 10 for being 10 being most stressful. So Greg, take over and want to explain the graph.
This right here is a significant part of data because, as you can see, before we showed the spider, his beats per minute was actually at 86, but when the spider was introduced right here around 246 seconds, it actually went up to 107. And this stimuli is very important because seeing data like this actually shows how effective the certain stresses are that we introduce the subjects to.
All right, and just a few more things. Another interesting fact we found was we had 2 balloon pops throughout the experiment, and the first balloon pop would always catch the subject off guard, and they would usually rate it pretty highly on a scale of 1 to 10. And the second balloon pop followed the ice bath, and the participant would either not notice the balloon pop or they would have much of a less reaction compared to the first one. And we think this has something to do with either being alert with your hands in the ice bath, or maybe your body still hasn't adjusted back to normal after that physiological challenge. Anything else?
We had a lot of different interesting findings, and we would love to look in the data more and try to find more trends. But we had a lot of fun testing, a lot of interesting data to look through. It was a great time.
Thank you.
Thank you.
Hi, I'm Sophia Young.
I'm Rebecca Denton.
I'm Sarah Larange. Our experiment is based off of the themes of conformity and race. We based our experiment off of a famous study conducted by Solomon Ash. What he did was he had about 6 people in a room, and he had 5 people we call plants, and he had a person who was like the subject. And what he asked everybody in the room participating was, which one of the lines on the left matches the one on the right? The 5 plants then answered incorrectly, and then Solomon Ash studied the subject to see what they would do. Would they conform with the group and answer incorrectly, or would they say the correct answer knowing that they were truly correct? What Ash did find out was that 75% of those subjects did conform to the group and answered incorrectly. What we did for our experiment was we turned it on its head and asked what was the influence of race on the study. So we had 2 different groups, a racial in-group and a racial out-group. The racial in-group being if you were the subject, you were the same race as the people in the room. If you were the subject in the racial out-group, you were a different race than the people within your room. We did the same methodology as Ash did, and with that methodology, through our experiment, we found out that in the racial in-group, the rate of conformity was only 10%. And then for the racial out-group, the rate of conformity was only 15%. You can tell from our data. Oh no, sorry. Keep going. Okay. You can tell that from our data.
Do you want me to start over?
Could you like?
Well, now I'm talking.
I'm sorry. I cannot think of the word at all.
Just keep going. We'll have to crop the video.
Okay, can we crop it? Okay.
Oh yeah, we can do that.
Okay, cool. Okay. You can tell from our data versus Solomon and Ash's data that there was about a 60% discrepancy between the rates of conformity.
So we tested a total of 60 students here at Governor's School East in our 3 trial groups. So we had a representative sample, which was just a random sample out of all the students here. We had the racial in-group, like Sophia said, where the subject being tested was the same race as the plants who knew what was going on in the room. And we had a racial out-group where the subject being tested was of a different race. So we have some of our demographics here for the subjects that we tested, including gender identity, area 1, and their racial distribution. So for our results, we tested not only the rates of conformity within each trial, but also the time that it took for each of the subjects to answer, so including all hesitations and pauses. What we found from our rates of conformity was that our racial in-group in comparison to our representative sample were both about 10%, however, the racial out-group was 15% in comparing to the representative sample of 10%. And our average time responses for the racial in-group and the representative sample, they were about the same. There were some slight negligible differences. However, the racial out-group trial had a much larger responding time and hesitation, whether it was the right answer or the wrong answer. And so our statistical analysis of these results does show that the larger gap for the racial out-group is due to a factor in the study and not just random chance.
So our findings do point to an influence of racial setting on an individual's behavior to conform, particularly if the subject is of a different race than those who are the plants in our experiment. So in analyzing our results, it's important to compare them to these results of the original ASH study and ask why there was a 60% difference between our results and the original study. So one reason for that might be that our experiment occurred around 70 years after the original and analyzed against the historical context of the 1950s and today. It's an important factor to consider. Additionally, our experiment did not use a random sample of people in the United States. Instead, we just tested people at Governor's School who may be inherently less likely to conform. Lastly, although we did control for students who had an in-class psychology education, other students may have known about the experiment from other sources. Well, despite those limitations, studying conformity in settings like these and with factors like race, gender, other things is important because the topic of conformity is socially significant and has lots of implications. 2 specific studies that point to implications of this are one by Chirwanka and Hansen that finds that conformity is a leading reason why teenagers engage in unsafe sexual activities. Additionally, a study by Mansfield et al. Finds that conformity is the reason that men are much less likely than women to ask for mental and physical health care even when it's necessary. Those are just 2 specific examples. Thank you.
Thank you. Hi, I'm Kelsey Patel. I'm Katrina Hugh. I'm Mackenzie Cochran. And I'm Rachel Graham-Eagle. And today we're going to talk to you about our project called Antibiotic Bacteria. So we all know how it typically goes. You use antibiotics to treat bacterial infections, and bacteria typically fall victim to them. But we wanted to study the opposite effect, where bacteria can actually be used to break down antibiotics and use them as fuel sources.
So how does this work? If you look here, the antibiotic that we're studying, penicillin, is actually a pretty simple molecular compound. Bacteria can break this compound down using various enzymes into simple carbon chains that they can use to build things. How this works is there are 3 sets of enzymes. You need a beta-lactamase to break down the beta-lactam ring in the penicillin. This is the dangerous part of the penicillin that pops the bacterial cell membranes and causes them to die. After the bacteria is able to neutralize the penicillin with beta-lactamase, it uses amidase to further break down the penicillin in half, and then a third phenylacetic acid catabolon to break it down into even smaller parts.
So how much antibiotic-eating bacteria is really out there? A study done by George Church found that bacteria were able to survive solely on penicillin G in all 11 of the diverse soil samples that were taken. Now this came as a surprise to us, so we wanted to see if we could say the same about samples of Meredith soil.
So what we did is we went around Meredith campus to 5 different locations and got soil from each location. We took that soil and extracted bacteria as seen in that picture, and put the bacteria in nutrient agar as well as different concentrations of penicillin. We already knew that because bacteria normally feeds off this nutrient agar, it will grow. However, we also hypothesized that after feeding off the penicillin, the bacteria will also grow. However, when we looked at the petri dishes, although the bacteria grew with the nutrient agar, it had no growth of the penicillin. This made us conclude that soil bacteria was either killed by the antibiotics, did not have the mechanisms to convert that antibiotics into food, or had mechanisms, but we were not able to see them with a microscope.
So even though we didn't find any antibiotic-eating bacteria in our soil samples at Meredith, we were still curious as to why there were so many antibiotic-eating bacteria showing up in other samples around the country. So the reason for this is because beta-lactamase and amidase genes are closely together in the bacterial genome. So when one is found, you'll often also find the other. In addition, the third part of the equation, the phenylacetic acid catabolon, is found in about 16% of all bacterial genomes. Therefore, taking into consideration the trillions of bacteria found in soil, it's not unlikely if the coincidence of all 3 necessary components occurs together.
So the process of the antibiotic-resistant bacteria clearing the environment of any pollutants is known as bioremediation. Now this can be used in hospitals, waste, and in farms as drug runoff. However, there are some negative aspects of this. You see, the bacteria have the ability to pass on the genes needed for antibiotic resistance to one another. So there is the possibility that a dangerous microbe can develop a resistance to the antibiotic meant to kill it. And there's also the possibility that the bacteria are only able to break down the penicillin and be completely useless against other drugs.
But the fact that these bacteria can break down penicillin in such small parts means that scientists can actually use this to splice and make new penicillin or other drug compounds that are semi-synthetic and that will work better against bacteria, or that bacteria are less likely to make a resistance to or break down.
Thank you.
Hi, I'm Sophie.
Hi, I'm Nidhi.
Hey, I'm Melanie.
And I'm Asta. So for our project, we wanted to transform some bacteria, so we used E. coli as our bacteria. We wanted to transform them by giving them the P glow plasmid. So on this plasmid is the GFP gene, which produces a green fluorescent protein. So any transformed E. coli can become fluorescent when they have the catalyst. And also on the plasmid are genes for ampicillin resistance. So if the E. coli are successfully transformed, they will become resistant to ampicillin. So we transformed half of the bacteria, half we didn't, and then we spread them out on 4 different plates. And in order to do the transformation itself, we used a series of heat shocks and ice shocks. So the heat shocks were to open up the plasma membranes of the E. coli to allow the gene to go into the E. coli and become incorporated into the genome. And the ice shocks were to help seal up the plasma membranes after. And then after that whole transformation was when we spread them out on these 4 plates.
So for the 4 plates, 2 of them didn't get the PGLO DNA, so they did not transform. The first one grew like normal, but for the second one, we gave it ampicillin, and the bacteria died since it was not transformed. For the other 2 ones, we did give them DNA. So for this one, we gave it the DNA and the ampicillin, and because it transformed, the bacteria survived. And for the last one, it survived as well because we gave it the DNA, but we also gave it something called arabinose. Arabinose is the catalyst for the GFP gene, which produces a green fluorescent protein that gives this plate the ability to glow.
So this is the plate, the arabinose, and the DNA, and I will show you how it glows.
Also, we had to do a lot of research before and after this process. We had to look up the GFP and what it was, and we also looked into CRISPR and why cutting out certain genes were, I just blanked, I'm so sorry.
Yeah, I was like, grow.
Really
grow.
You can keep going. We can just drop that there.
Yeah. So basically we had to research why certain DNA, if you cut the genes out, why they are affected and why adding some give it like the flush, it's like ours does. We also wanted to look into why certain GMOs are healthy for some people and why people believe that they aren't healthy, such as like human side effects. That brings us to the research link between IBS, irritable bowel syndrome, and effect of GMOs. A lot of people believe that GMOs cause IBS. However, since IBS is brought out such through a long period of time, and the conditions are as well, there are so many other factors such as your environment that could influence IBS. So there's no actual real connection between IBS and GMOs, and also benefits and benefits.
So I did my research on detriments, benefits, and ethical issues. So for some detriments, it was said that GMOs can cause reactions and stomach problems such as IBS. However, when we did more research, we found out that there was no relationship between the two, and there was more of a correlation rather than a causation. And some benefits are that it's more nutritious, tastier, and then it also helps with production so that it can go to other countries that need more crops. Now in this picture right here, you can see that on the left column, the corn looks really nice and healthy. And that's the BT corn, the corn that received the genetically modified organism. And the ones on the right are the ones that are not BT corn, which means that they did not receive the GMO. And you can see that they look pretty rotten and infected by pesticides, which got us thinking that maybe GMOs are actually very helpful to you. Now, there are some ethical issues that have been brought up, such as it's a threat to the organisms that are receiving this injection of GMOs, and that it's a threat to the environment and ecosystem, as GMOs do have another detriment that they produce toxins and antibiotic resistance, so through cross-pollination, this toxin can get around and cause death of many plants and many plants. Now, there's another widely known ethical issue such that scientists who are doing this injection of GMOs are kind of going above what God naturally created and manipulating it and making it into something man-made. However, through our research and our experiment and breaking down our experiment, we came to the conclusion that GMOs might actually be leading us to a bright future. Thank you.